Welcome to Easy Microbiology.
Good morning, everyone. Today, I'm going to talk to you about the highly virulent subspecies of the gram-negative bacterium Francisella tularensis subsp. Tularensis. This bacterium is primarily found in North America and is associated with animals such as rabbits, hares, and rodents. Let me begin by introducing the importance and implications of this bacterium. Francisella tularensis subsp. Tularensis, or type A, is a highly virulent subspecies of the gram-negative bacterium F. tularensis. This form of bacteria is identified as the most pathogenic type among its variants and is primarily found in North America. It can cause severe disease manifestations in humans, as well as in animals, such as rabbits, hares, and rodents. F. Tularensis subsp. Tularensis is a bacterium endemic to North America, predominantly in the USA and Canada. This highly virulent subspecies resides in animals such as rabbits, hares, and rodents, which serve as a source for the bacteria. Francisella tularensis subsp. Tularensis is a small, non-motile, pleomorphic bacterium classified as gram-negative. Under a light microscope, it is not easily distinguished as tiny, cocobacillary-shaped cells, measuring only 0.2 to 0.3 micrometers. Though small, this pathogen is capable of infecting humans and wild and domesticated animals, thus being categorized as a Category A pathogen by the U.S. Centers for Disease Control and Prevention. F. Tularensis subsp. Tularensis can be challenging to culture due to its fussy requirements. Nonetheless, cysteine heart agar and modified Thayer-Martin agar, both specialized media, can provide the optimum conditions for the bacterium to proliferate. Diagnostic testing can be done on the appropriate culture medium to verify the presence of this subspecies, thus ensuring a successful cure of the infectious agent. F. Tularensis subsp. Tularensis is an oxidase-negative, non-hydrogen sulfide-producing, and non-fermentation bacterium. Its metabolic capabilities are very limited, making it metabolically inert. As such, it is a very difficult bacterium to research and understand. However, its biochemical characteristics have been studied extensively, and we are now able to better understand this organism and its impact. Humans can become infected with F. tularensis subsp. Tularensis, a species of bacteria that infects small mammals such as rabbits and rodents, when coming in contact with infected animals or inhaling contaminated aerosol. Additionally, infection can occur through ingestion of contaminated food or water. To reduce the risk of infection, people should be aware of the dangers and take precautionary measures. F. Tularensis subsp. Tularensis is a highly virulent bacterium that is found primarily in North America and associated with animals such as rabbits, hares, and rodents. It possesses several virulence factors that contribute to its pathogenicity. These factors include lipopolysaccharide, capsule, and various proteins that are involved in invasion, replication, and immune evasion.
F. Tularensis subsp. Tularensis is generally susceptible to multiple antibiotics. However, sporadic cases of antibiotic resistance have been reported, mainly through genetic mutations or the acquisition of resistance genes.
Epidemiology is the study of the occurrence, transmission, and control of diseases. Francisella tularensis subsp. Tularensis is a very infectious form of the gram-negative bacterium that is commonly found in certain parts of North America. It is mainly found in places where people come into contact with infected animals or the places they live, such as rabbits, hares, and rodents. People can get the disease by breathing in bacteria-filled dust, eating contaminated food or water, touching animals with the disease, or getting bitten by infected ticks, deer flies, or other insects. It is therefore important to be careful when dealing with animals or their habitations in affected areas. F. Tularensis subsp. Tularensis pathogenesis is complicated and multifaceted. It can enter the body through ingestion, inhalation, skin abrasion, or through arthropod vectors and generally infects macrophages and other immune cells. It can bypass the host immune system, replicate within cells, and spread systemically, usually causing a serious systemic illness. F. Tularensis subsp. Tularensis is known to trigger both innate and adaptive immune responses in the host. While humoral immunity is important to clear the infection, cell-mediated immunity, specifically T-cell responses, has been shown to be a critical factor in controlling tularemia. Studies have shown that successful resolution of infection from F. tularensis subsp. Tularensis is dependent on the activation of vaccinations. This highlights the importance of understanding the pathogenesis of the bacterium and its pathogen-host interactions. It is also important to note that increased immunization against F. tularensis subsp. Tularensis can reduce the morbidity and mortality associated with the disease. The diagnosis of F. tularensis subsp. Tularensis infection involves several methods, including culturing, PCR, and serological testing. However, due to the high infectivity of this highly virulent subspecies, it is important to take the necessary precautions and use specialized biosafety protocols when dealing with such samples. To prevent tularemia caused by F. tularensis subsp. Tularensis, it is essential to reduce the risk of infection. Avoid contact with infected animals or their carcasses and use protective measures when handling potentially contaminated materials. Wearing appropriate clothing and using repellents to prevent arthropod bites can also be beneficial. Unfortunately, there is no available vaccine for humans against type A tularemia. Antibiotics are the most common form of treatment for tularemia caused by F. tularensis subsp. Tularensis. Many drugs, including streptomycin, gentamicin, doxycycline, and ciprofloxacin, have been used successfully to treat this potentially dangerous infection. Prompt treatment is vital for the best outcome, so it is important to seek medical attention as soon as possible after diagnosis. Based on our research, type A F. tularensis subsp. Tularensis is a highly virulent subspecies of F. tularensis that is found primarily in North America and is linked to severe cases of tularemia in humans. We have learned that understanding its characteristics, epidemiology, pathogenesis, diagnosis, prevention, and treatment is critical for the effective management and control of this particularly pathogenic subspecies of F. tularensis. We hope that this presentation provided key insights into the topic and appreciate your time. Thank you.